Cool. Right, okay, so there were quite a lot of sessions, but only taken half of it. Or part of it, yeah. So the session kicks off with Professor Will Jacobs of Princeton University to tell us how to design multiphasic materials. So thank you.
Okay. Thank you, Richard. Thanks to the organizers for the invitation. All right, so it's my pleasure to tell you about some of our recent work thinking about multi-component systems and how we can design them. The main thing that I want to emphasize today is that a special feature of multi-component systems is that you can make many different phases or many different structures, all from the same set of components. This happens in many realms of science. One that we've already heard about this afternoon is biomolecular condensates. So these are structures that form without membranes inside of living cells. They form via phase separation, and as already pointed out, in many cases you get these beautiful structures that involve many different phase-separated fluids. Here it looks kind of like these Matryoshka dolls that were mentioned earlier. This is a classic example, the nucleolus. This is where ribosomes are produced inside of nuclei, and each one of these compartments that are color-coded here has a distinct chemical environment, is involved in a distinct part of the ribosome assembly pathway. Right? But it's not just when we have these interlocking multiphasic structures. We also have a wide variety of different condensates that have been characterized at this point. The key point here is that they are in the same environment, but they are immiscible. They're chemically distinct. They're highly complex. If you look at what is partitioning into a condensate versus the rest of the cytosol or the nucleosol, we have a large number of distinct gene products here. And this isn't a simple one-to-one, protein A goes into phase alpha. We can have examples where certain proteins are partitioning into multiple distinct condensates that are nonetheless immiscible. Right? And this is something that people have taken a real interest recently in trying to design. You can create multiphasic structures with disordered proteins, nucleic acids, entirely synthetic polymers. All of these examples are trying to get at this sort of emergent organization that we see in biology, but we're nowhere near the level of complexity of what we have inside a living cell. It was also pointed out a couple of years back that in this paper by Arvind Murugan and Zorana, that you can get the same sort of emergence of many different structures from a single set of components if you think of the subunits as being puzzle pieces that can interlock in different ways. This was beautifully shown in a recent paper with DNA origami, that this can actually be done in practice. And in terms of colloidal self-assembly, we're really getting to the point where I think we do have the ability to design truly complex interaction matrices, all between different types of subunits that could give rise to many different crystal polymorphs that are all encoded from a given set of subunits. And the structure that emerges depends on the initial seed that is placed into a solution. Right? So this is an emerging direction. In all of these cases, there are a couple of theoretical questions that come up. First is, if you wish to design such a system, how many distinct structures or phases in biology can you actually encode in a given set of components? And in order to actually achieve that in practice, how do we design the molecules or colloidal particles that would be in this mixture? And then secondly, if we want to actually have a particular one of these encoded structures or phases emerge, then how must we control the assembly process? This is something extremely important in biology because these condensates tend to emerge for the most part under nucleation and growth pathways, so the initial nucleation condition is very important. Okay, so in the first part, I'd like to tell you about some ways of solving this inverse design problem. I pose this as inverse design because when we're thinking about a very high dimensional parameter space, it's very useful to have a particular goal in mind and work backwards as opposed to hunting around with random parameters. So the paradigm here what I'd like to propose is thinking about this as a programming problem. So we are going to specify a set of phases that we would like to have emerge, and then we will work backwards to figure out whether this is possible and what the interactions would have to be. Okay? So just to give you a cartoon of what we're trying to do here, imagine we have a mixture of many different components. These little blobs here represent different molecules, and I wish to encode different condensed phases. And a phase will be defined by the subset of these species that comprise the majority of the molecules in that phase. So this would, say, be phase alpha. I could have a second one encoded to make this problem interesting. Maybe the same species appears in multiple different phases, but again, these should be immiscible, and so on. So this constitutes my design problem. And then the goal here at equilibrium should be to design a free energy landscape that gives me a local minimum at a point in composition space that corresponds to these design targets. Okay, so to get started, we can use a mean field theory to facilitate this inverse design. Okay, so we're just going to use the standard Flory-Huggins model. The key thing to focus on is that we have this matrix of pairwise intermolecular interactions, and this is the thing that we wish to design. Okay. So we have two types of constraints in this problem. One comes from the fact that we are, by choice, prescribing the precise compositions of the phases that we would like to stabilize. But then, of course, we also have to deal with thermodynamics, right? So to have local minima on this free energy landscape, we need to end up with locally equal chemical potentials and osmotic pressures, but also a positive definite second derivative matrix. Right? And then we want to do this and solve for epsilon. So this ends up being a challenging nonlinear problem, but it's possible to do a relaxation of this through some well-controlled approximations. And essentially, if you think about this very high-dimensional space of interaction matrices, we are going to try to solve for a slightly larger region, which will allow us to prove whether a particular set of phase compositions is possible or not. Okay, so before I show you these results, it's useful to think about a heuristic. So here, if you take a particular limit of that problem and think about this second derivative matrix, it's useful to write down what we call a target graph, which is just this adjacency matrix that tells us in our design problem, we put a 1 if our species I and j are both supposed to be enriched in high concentration in any particular phase. That gives us this matrix. And one can show that in this particular limit, this design problem is infeasible if it just so happens that that any of our target phases end up being embedded inside of a clique of a larger size in this matrix. Right? So this is not actually solving the thermodynamic problem fully, but it gives us a nice way of estimating when we're going to be able to solve this problem at all, because we can use random graph theory to make some scaling arguments based on different clique sizes. And so the scaling prediction that comes out of this is that you should typically be able to find a solution that satisfies these constraints if the number of phases that you wish to program or encode is smaller than something that scales in this way. Right? So again, I'm putting a select subset of m components in each one of my distinct phases. Right? So indeed, if you do the numerics using this convex optimization approach, you can find that the typical number of condensates that you can successfully encode with such a free energy landscape scales as a power law as a function of the number of components, and this scaling exponent increases as you increase the complexity of any given condensate or any given phase. So this is the number, capital M, of components that I'm putting into each condensate. And all of this follows the scaling prediction quite well. Okay? And so if this is surprising at all that you can get a super linear growth in the number of phases with the number of components, seeming to maybe contradict Professor Gibbs, there's not actually a problem here. We're thinking again about local equilibrium, and we're simply trying to estimate the number of local minima on this very, very high-dimensional free energy landscape. Right, and I'll show you in a little bit that this actually does work if you perform more detailed simulations. Right. But at this point, we only have interaction matrices, which are very nice in a Flory-Huggins sort of framework, but that doesn't tell us how we actually get these, how we would actually design real molecules, or what the more chemical rules of the biomolecules that form intracellular condensates would have to be. Right? I should also emphasize that this convex optimization approach gave us constraints on these interaction matrices, but it didn't tell us that there was a single one. In fact, this is often a highly degenerate problem. So we should be looking for solutions to the problem that are the simplest. If we stay within this mean field framework, then it's possible to factorize this epsilon interaction matrix and break this up into linearly independent features. So for a concrete example, I'm going to talk about designing heteropolymers. And if we're in this heteropolymer case, then our matrix U here would represent the interactions between the different monomers that are built into heteropolymers, and the W matrix would tell us the count encoding, or the frequency with which we see any particular monomer in one of the heteropolymeric species. So if we're looking for the simplest molecular representation of these interaction matrices, we should be trying to minimize in general, the rank, right? So this is a measure of the complexity of these interaction matrices. And we wish to know, for a particular design problem, trying to encode these compositionally distinct phases, how many of these features would we actually need? All right, so this is a project that a graduate student in my group, Fan, took on. And so again, our basic idea is we wish to prescribe some particular set of phases with precise compositions. We're going to do this optimization process using convex optimization tools, but we're going to specifically regularize to find the lowest rank matrix. And that will tell us something about the minimum complexity, or in this case, since we're constructing heteropolymers, the number of monomers that we would need. In particular, it tells us the number of monomers of each type that should go into a polymer, and then we arrange them in a way that makes them as non-blocky as possible in order to give our mean field theory the best chance that it can have to work. Right, so we have this design pipeline that now says, if I want to make this, I need molecular building blocks that are at least this complex, and here is a proposed design solution. Right. So now we can turn to molecular dynamic simulations and see whether our designed phases are indeed immiscible. Again, this is non-trivial because in the design problem, some of the chains are supposed to be partitioning strongly into distinct phases, but the phases shouldn't mix. And the chains also are sharing a limited number of monomer types. But indeed, this does work if you define an order parameter that simply tells us how close is the composition that I observe at some point in my simulation box to the requested composition in my design problem. Then we can see that these phases indeed match the targets because the order parameter is 1, aside from some interesting interfacial effects. And so in this case, we need to look at 3 simulations to look at these combinations. Okay, so you can ask, what is this actually the simplest possible solution that would create this multiphasic structure? And so according to this mean field theory, you should be comparing sort of the error that's introduced by reducing the number of monomer types to something about the stability of your free energy landscape. This number should be small, and indeed, we can get away with only using 3 monomer types in this way. All of these phases end up just mixing together for any design of heteropolymers where we only used 2 monomer types. Right, so this gives us some assurance that we are going in the right direction for looking at the molecular rules for being able to build these multiphasic structures. Okay, so you can take this, you can make much more complex systems, but the key takeaway here is that this inverse design approach can give you a way of not only saying whether a particular multiphasic organization is feasible, but also how complex the molecules you would need in order to actually achieve that. All right. So far this is just about the equilibrium properties. I also mentioned that we might want to control the growth of these structures. And so the way that we're thinking about this is imagine that you have a mixture, again, where I've designed the system. In this case, there should be approximately 100 encoded phases. You only see blue and red because well, 100 colors would be unmanageable. But in particular, red components here are all of those that are designed to be enriched in this particular seeded phase. Right? And so not only should this be stable at equilibrium, but if I insert this into this supersaturated mixture, it should be able to grow without changing composition, and that's what you see here. And indeed, if you go and check this for all of the designed phases, you can retrieve each one selectively with a seed, and again follows this sort of scaling prediction that came out of this graph theory. Okay. So we wanted to go a little bit farther and understand exactly under what conditions this retrieval process is possible in these sorts of systems. And so we can go back to a simple mean field model where we're thinking about when particles or molecules are incorporated from a supersaturated vapor phase into this bulk condensed phase. Everything interesting is happening at the interface. And so you can define the growth rate of one of these phases as essentially what's coming in from the vapor phase and what would leave through spontaneous detachments. And then I can solve for the steady state in order to develop a dynamical systems model of when this retrieval process would be stable. Okay, so I'm going to solve for this steady state growth. This is what's happening at the interface, and this represents the fact that the interface is moving forward in this growth direction. Okay, so if you do this, you end up arriving at a dynamical phase diagram that tells you about the conditions under which you can selectively grow these encoded structures. Point out two key parameters. One has to do with the interaction between each one of these so-called ordered phases, or the encoded ones, versus just having a random mixture of the molecules in a condensed phase. And this one is essentially the stability of this random phase that could possibly grow. And I want to draw your attention to a particular regime that I think is quite important and also quite surprising. This orange region here, labeled dynamical transition, is where we get a bifurcation diagram that looks something like this on the right. Essentially, what it's telling us is that we have a possibility for two different types of phases to grow. One would be any one of our ordered or designed structures, where this is that would be the seeded state. If we're in the blue regime here, by controlling the chemical potential in the vapor phase, we will have a positive growth rate. But you could also get your disordered state to grow at some other chemical potential, and there is in principle a crossover between these two, where I could have a dynamical coexistence of two different types of growing structures. Okay. So to try to see if this dynamical coexistence can actually be seen in a simulation, we turn to a slightly different model of multi-polymorph assembly. So here we're encoding structured phases as opposed to just liquid-like phases. But the key idea is that by introducing some directional interactions, you can now use permutations of the subunits as opposed to just composition of subunits in order to encode very many distinct structures, where the overall interaction matrix is just given by the union of all of these encoded designs. Right? So this is inspired by, again, this wonderful paper from Arvind. But here we're looking at multi-component crystals, so we can see steady state growth and look for a signature of this dynamical phase transition. Right, so if you take this picture and you map it back onto our mean field model, it says that as you increase the number of encoded structures, at some point you leave this orange regime here, enter this regime where you have no possibility for ordered growth. Right? So that defines the capacity of the system. This is, in this case, the maximum number of structures that you could encode, predicted to be 6 in this case. Right, so that's the mean field prediction. If you actually do kinetic Monte Carlo simulations, we can create this assembly diagram that tells us the conditions under which you can get stable seeded growth. So again, what I'm doing is I'm inserting into the supersaturated mixture a particular seed of one of the encoded structures, let's say unit cell number 1, and then I'm watching it grow. As the simulation progresses, the interface will move to the right. So this green region here, for different numbers of encoded structures, tells us about where we're able to precisely retrieve this structure and build either a phase or a material that is defined by the seed. The blue line tells us coexistence, so this would be where everything is in equilibrium. And so we want to look at how far essentially we can supersaturate beyond that and still retain this seeded control. Right? So there are a couple failure modes here. One possibility is that there's homogeneous nucleation. We can also have heterogeneous nucleation of different encoded structures that, that grow on top of one another. But what I want to draw your attention to at this storage capacity is the fact that in this carefully chosen example, you can actually see a direct transition from the growth of one of these encoded structures to this disordered phase here. So what exactly does that look like? Indeed, we actually do get a crossover of these two different growth velocities. So our ordered growth is very slow and relatively insensitive to the supersaturation. But the disordered growth is highly dependent on this, and there's a crossover where both structures are growing at the same speed. If you are at a lower supersaturation, we have this stable, steady state retrieval. If we go far beyond, then we get a rapid takeover of this disordered structure. But I think quite interestingly, if you're right very close to this transition point here, then you can get what looks like this wetting layer, except everything here is in steady state. This layer fluctuates, and if you monitor its width, as you approach this dynamical transition here, where we have coexistence between two different growing structures, this width appears to diverge. Right? So this has a lot of signatures of a first-order-like transition, where we're only able to see it if we're very, very close to the storage capacity where this is the dominant failure mode. This sets in before either those heterogeneous or homogeneous nucleation. All right, so the key takeaway here is that we can indeed show that for some of these multiphasic models, a dynamical coexistence is what limits your ability to retrieve seeded structures if you are very close to this storage capacity. Okay, so to sum up, what I'd like to emphasize here is that in multi-component systems where we have the ability to design interactions between different molecules or colloidal particles, you can get a surprisingly large number of distinct phases, and all of this can be programmed. That is, if you have the ability to completely and independently tune all of the elements of your interaction matrix. Right? In practice, the number of independent molecular interaction modes tends to actually limit what you can achieve because of your limited ability to design all of the interaction elements of this matrix independently. Using inverse design techniques, we can predict the feasibility of a system, compute the required number of interaction modes to stabilize it, and use this for rational design processes. And we have the possibility of different failure modes when trying to retrieve particular encoded structures via nucleation and growth pathways. All right, so I think that as we go forward, these ideas may prove to be very useful for guiding the design of multifunctional materials, either very close to these sort of existing biological systems, or some ways that are more based in programmable self-assembly. Right, so I'd like to thank you all for your attention. Point out again, graduate student Fan, who did most of the simulations that I showed you, and acknowledge funding support. So thanks.
Lots of other questions. Questions for Will? Well,
just a quick one. So let's say you have your different interaction modes, you just set the number of them. I suppose that in the real world, there's probably some crosstalk. They don't really separate, but there's some limiting or element of English as well as the number of modes. So if you think about sensitivities and crosstalk on the modes, and kind of
so crosstalk, I mean, so if we take the example that I talked about with these monomer interactions, right? So each monomer interacts with every other monomer, right? You can have zeros, but you might not, right? That's not the key thing here. So we're not saying that this inner matrix would be diagonal or have any particular structure. We're only saying that that the rank of the overall epsilon matrix is constrained. Right? Right. So of course, you still need to have truly distinct interaction modes, right? Like you could, of course, have a negligible interaction that trivially increases your rank, right? So of course, there is also a limitation on that. In the simulations that I showed here, we had complete control over these interactions, and of course, in something like a DNA functionalized colloidal situation, you could achieve that. If you have a large enough library of ways of interacting, yes, of course. But absolutely, there is one additional layer of constraint on like what nature gives you as that interaction matrix that is not considered in this problem, but we are working on it.
Can you use your methods to understand your those failures? You can also suggest ways to change the molecules so that they won't fail.
A tough question.
Can we do it? Yes. Confidently. Absolutely. I think one of the key distinctions here is that we're thinking mostly about this as an inverse design problem and trying to put them in by design. Of course, the prerequisite for doing this when you haven't designed them, but they're there because of nature, is that you have to know what they are, what their relative stabilities are. I think what I showed you in that particular dynamical mean field model assumes that everything has, that all the different polymorphs have similar stabilities. But in principle, yes, I think that a lot of these ideas about different failure modes and when they would appear, and depending on the number of polymorphs that exist, should indeed carry over. Thank you.
I have a detail. Then if you look at the sequences, often two flavors per chain, and then you have multiple chains with different flavors in one. So that's another optimization that you like. I can certainly imagine that you would get condensates if you just have two flavors, if you play around nicely enough with block copolymers.
Absolutely.
So why is it that you said two? And what's the extra asterisks on the 3 and the 4?
We haven't been able to do it with two. Strictly speaking, this this rank that we've calculated is an upper bound. And this is also why I think it's a bit better to describe this as more abstract feature interactions as opposed to always explicitly monomers. The reason being that indeed blocky sequences that we explicitly didn't look at here give you additional types of interaction modes that are sort of orthogonal from just composition. So indeed, it may well be possible to make that particular set of phases with those prescribed compositions using even fewer monomer types, but I think you would still need 3 interaction modes. It's just that maybe one of those comes from sequence patterning.
Got it. Okay. So
We did to make direct contact with that mean field theory, but I think that the blockiness is actually a tool that should be used And again, we're thinking about that.
When talking about superlinear capacity, in these networks, I'm immediately wanted to think about Hopfield network with high order interactions. So I'm wondering if you can map what you have, it seems like two particle interaction to a higher order model, giving superlinear capacity.
Right. I think one key thing is that in typically when we think about the Hopfield models, we think about all of the spins being programmed in each encoded state. Whereas here, that's not exactly the case, because we have, in each condensed phase, there may be a large number of components that are enriched, but it's not all of them. Right? So it's a slightly different problem there. We, strictly speaking, only have pairwise interactions. It's a nonlinear Flory-Huggins model, but there's only pairwise interactions. So I think the distinction with a Hopfield model comes from that fact, that not every component is in every phase. And that's where the superlinearity comes from.
The final question, sorry.
Sorry, a question about surfactants. Do they change the map entirely, stabilize more phases if you start to surfactants?
We haven't thought about that so carefully in this particular case. I can point out one thing is that if you've noticed, because the design problem is only talking about bulk phases, there was no control over what the interfaces did. And so if you look close to the interfaces in those MD simulations, they don't match what we might have thought. And I think that there's additional ability to control that sort of thing, obviously, with surfactants. But whether they give you entirely new bulk phases, I would probably not expect so. Yeah. Definitely. Yes. Yes, absolutely. In what way affect, right?
Well,
Like, absolutely. Absolutely, yes. Yes. And I think that especially in the biological cases, designing things to have particular chemical compositions at the interfaces is super important, right? So that, right, yes. So indeed, the key thing here is that that was just not part of the inputs to the design problem, but one should do that.